record the session. And thank you everybody for joining this webinar. So before we start, I would like to tell you about the captioning. You have a real person that is doing the live captioning. So it's not done by Zoom, it's a real person. So hopefully the captioning will be very high quality. And you can also ask questions. You have to click on the button Q&A and you can type in your questions and we will try to answer to your questions at the end of this session. So thank you for joining again. And if you would like to watch the recording, I'll tell your friends and family to watch the recording and we'll watch it again or watch the previous webinar. Please sign up, subscribe to our YouTube channel. You have the link here on the screen, but you can also use your phone and use this QR code in order to reach immediately that our page, our YouTube page, and then do subscribe. And then you will have this recording as well. My name is Gilles Atlan. I'm the father of Karen. She's 15 years old and she has NF2. And because of her, I'm here as Vice President of NF2 BioSolutions. So I'm right now in Los Angeles with Dr. Brenner, and tomorrow both of us will present at the NF2 State of the Art conference in UCLA to give an update to the NF2 research community. So 4 years ago, when we started the patient-led nonprofit NF2 BioSolutions, at that time, Dr. Gary Brenner was one of the very few researchers with innovative approach to tackle NF2. And his lab at Harvard Medical School was the first lab that we founded, thanks to all of your donations and your support. And please continue to support us, NF2 BioSolutions, so we can give a better future to all the NF2 patients. Gary Brenner is also a pain medicine physician at Mass General Hospital, treating patients that are suffering from rare disorder. So thank you, Gary, for making the time today to be with us. And on the East Coast, we have here Dr. Neil Kirby in Boston. Neil is the CEO of Mulberry, working on developing the drug that emerged from Dr. Brenner's lab. Neil has more than 20 years experience in drug development and proven success in developing rare disease drugs. We feel very lucky to have Neil, and we know that Dr. Brenner therapy is in very good hands, and we trust that Neil will succeed. One example of this trust is that one of Neil's previous colleagues, Tal Fuhrer, based in Israel, invested $1 million in Mulberry together with many more angel investors. So this simply shows the trust that people have in Neil and his execution capacity to bring the drug to the clinic. So without further ado, let's start the drug development update with Dr. Neil Kirby. Thank you, Neil.
Thank you, Gilles. Thanks for the kind introduction. It's a pleasure to be with you this morning. Gary, if you can share your screen, that would be great. You've got my slides.
It should be coming. And let me also thank you, Gilles, for the introduction and your support. And I do feel like I should make the disclosure that I'm a scientific founder and stockholder in Mulberry, which should occur. You guys should know that. And right. And this is true. It's really wonderful to have Neil driving this effort to get this product to human trials. I can't think of a better steward of the technology that we developed. So I really want to thank you, Neil.
Thanks, Gary. Appreciate that. So what we'll do today is to give you an introduction to Mulberry, because I think that you probably, a lot of people on the call don't know who we are and what we're doing. As well as Gary will give an overview of some of the work that's gone into our lead technology with the immunotherapy for Schwannomas. Gary, if you can click to the next one.
There we go.
Great. So this is our mission statement, and I won't read it out. You can read it at your leisure. We're really focused on NF2 to start with, and that's where we're spending the majority of our time, is developing the bacteria therapy that was developed and researched in Gary's lab and taking that to the clinic. Next slide. So we were established actually as a company called NF Bio, and I'll talk later about who the founders were. As Gary mentioned, he is one of the founders, as is Professor John Mekalanos from Harvard, who I believe also presented to NF2 BioSolutions in the past. We actually changed the name to Mulberry Biotherapeutics in February of 2021. And our first set of funding was actually from NIH, was an STTR grant for $700,000, which really got us started. In April of 2021, we were able to raise $2.1 million to allow us to proceed and really have a good jump start on everything that we needed to do. And I'll spend some time later describing exactly what we've been able to do on the development side as a result of the work that Gary will present on the research side. So we are completely focused right now on NF2. And it is obviously, leveraging the work out of Mass General on bacteria-mediated therapy. And I know that Dr. Brenner is going to tell you a lot more about that in his presentation. We're very fortunate to have Gary. I've worked with a lot of academic collaborators over the years. And I have to say that, it's not just because he's on the call. I think Gary is the best academic collaborator I've ever worked with. So we're very fortunate to have him involved. He'll work you through the data that he generated, and he'll tell you about the publication that was made recently, earlier this year, that describes that data. So we have a very, very solid scientific basis to move forward. And that's, probably the most important thing in developing a drug, is to understand that there's real potential here. And I'll talk more later about FDA, the Food and Drug Administration, and what the steps are that we need to take, as well as where we are in the development process. So I'm going to hand it over to Gary now so that he can work you through the data.
Thanks, Neil, and thanks for your really kind comment. If I start going too slowly, just shoo me along. My feelings won't be hurt. So I'm not going to talk to this group about the physiology of Schwannomas, other than to say that while there's not a great deal of information on the immune microenvironment or the immune milieu, and I should say that my primary scientific background is in immunology, there is some data to strongly suggest that these benign neoplasms, benign in that they don't invade other tissues and don't metastasize, but not benign, as you know, in what they can do, are what we call immunologically cold. It's not a very hot inflammatory environment that the immune system can see. And one of the things our lab's really always been interested in is manipulating the tumor or the host immune system in some way so that the host, in this case, the individual with the disease, can themselves attack the tumor with their immune system. And this has worked for some malignancies. So some of you may be aware that we tried to use a gene therapy strategy to turn on the host immune system and kill the tumors. And while we were able to deliver suicide genes and destroy tumors to some degree, we never activated the immune system. And I have to give Sharif Ahmed, my postdoc at the time, the credit for bringing to the forefront the idea of using bacteria. I looked at the literature. There's exciting data for bacterial therapy, and you can see that line there, that there's actually 2 centuries of observational data that say when people get a systemic bacterial infection, people with cancer, the cancer seems to just miraculously go away. And that was first noticed in France. I find this interesting. Some physicians treating prostitutes, and they observed that occasionally a prostitute with cancer when they got an infection and they didn't know what bacteria were, the cancer went away. And today there is an FDA-approved product for treatment of bladder cancer, and it's attenuated bacteria, and you'll hear more about that later. And the claimed cure rate is 60%. So it's pretty amazing. So why schwannoma and bacterial therapy? Well, we really felt that these non-cancerous tumors are ideally suited for bacterial therapy. I talked to you a little bit about the fact that they're cold tumors. I will just mention they have immune cells in them, such as macrophages and T cells that you may have heard about, that are in a suppressed state. And bacteria are known to be able to take those kind of cold, suppressed immune cells and turn them into very active immune cells in a pro-inflammatory environment. I will mention that bacteria, in fact, the bacteria we used has already been used in clinical trials, and it has shown to be safe with cancers. Now, they didn't show that they help the cancers, but we really feel that we have a completely different type of tumor, and we think that it will have efficacy for the schwannomas. One of the main features that I think will lead to efficacy, besides the fact that they're cold immunologically, is the schwannoma tumor cells tend to divide very slowly, and they're genetically stable. So they don't have the capacity to evade a new therapeutic, such as bacteria, in the way that a fast-growing, genetically unstable cancer would. We would intend to inject the product directly into the tumor, further enhancing the safety. And to kind of move to the lab information, I can tell you as an overview, we've demonstrated that this approach of taking these attenuated bacteria that have already been proven safe in human trials and inject them directly into schwannomas in two models, both a human, what we call a human xenograft model, where we take human tumor cells and they grow in mice that are immunologically suppressed, because if they're not, they just kill human cells, or a mouse schwannoma model, which grow in immunocompetent animals. So we have these two models, and I'll just tell you, clear efficacy. And I'll also say much better efficacy than we've ever seen with our gene therapy approaches. And importantly, as Neil alluded to, you need to know or have a sense of how it's working in order to really give you confidence that this can work in human beings. So what are the mechanisms of action? And this is, I won't get into this too deeply because it would take a couple hours, but it's really exciting. One is these bacteria, these salmonella bacteria, actually have mechanisms for getting directly into the tumor cells, replicating in the tumor cells and killing them. And we can clearly show that this occurs. We show that they get into tumor cells, and we show that once they're in, they can kill tumor cells by what's called programmed cell death or apoptosis, as well as something called immunologic cell death that leads to a pro-inflammatory environment. Interestingly, and this is, all these reasons are what we hope for and why we chose to work on this project, they're anti-angiogenic. All of you know about Avastin, bevacizumab, and that in some patients, it has real efficacy, the drug that is an angiogenesis inhibitor. Amazingly, these bacteria inhibit angiogenesis, and we've shown that that occurs in our animal models too. Thirdly, I mentioned these tumors have a lot of macrophages. That's an immune cell type. And there's different types of macrophages. Very globally, there's the M1 and M2 type. The M2 type, which most of them are in these schwannoma tumors in humans, are actually tumorigenic. They actually promote growth of tumor and inhibit immune activation. And what was known in the literature, and we showed in our Schwannoma models, well, known from the literature with cancer, is that the bacteria can shift these pro-tumor forming M2 macrophages to anti-tumor or tumor killing M1, and we show that that happens. And perhaps most excitingly, we show that there's an induction by the bacteria of host or the animal's anti-tumor adaptive immune responses. Now, you may know there's innate immune responses, which are critical, and that's kind of like the usual inflammation and non-specific killing, and adaptive immune responses, which are T cells and B cells that have to recognize proteins on an invading cell. And that means seeing a tumor cell as an invading cell and then specifically kill them. And the reason that's so exciting is there's the potential to control growth of multiple tumors following treatment of a single schwannoma, and potential for lasting anti-tumor immunity following a treatment. And I will tell you that we have data to support both of these. And the other thing that we've published and we were interested in is if we can use bacteria to kill tumor cells and initiate this immunologic response, can we use other treatments to maintain that? And we've shown that we can do that with checkpoint inhibition. And everything we're doing has a basis in what's known about these tumors in human beings. And I'll just quickly show you some data. This is all published. You can get it online. This is the human schwannoma that we implant into mice. And the black line is the untreated tumors. Again, we just inject bacteria, live attenuated bacteria, into the tumor. And I can tell you in humans, and like I'm a pain guy, I inject all sorts of things in the nervous system. We can locate these tumors with either MRI or ultrasound imaging and get a needle in them and direct them. Very straightforward. So that's why we're injecting directly. The black line is tumors that are control tumors injected with a saline solution. And amazingly, look at the green line. Those are the tumors injected with the bacteria. And what I can tell you is basically 75% of the tumors just go away completely. No evidence that they're still there. There's no other therapy that's had this kind of effect. And that's been replicated across multiple studies. This 75% is a number I'm quoting from 3 separate studies, which is important. This is the mouse schwannoma model, and you can see a similar effect. It's not quite as strong. And what I'll tell you is these human tumors, this is weeks. So they're growing relatively slowly, but still faster than what a human tumor would grow. And in these mouse tumor schwannomas, it's days. They're actually growing quite fast. I was blown away that we saw this kind of effect given how fast they're growing. And that's one of the reasons I really think this has a very good chance of working in humans, because the human tumors are growing even slower, giving the bacteria more of a chance to kill them and induce host responses. And I really talked about all of this, and I don't want to get into these data too deeply. I'll just remind you that these bacteria get in and kill these tumors by a number of mechanisms. One is programmed cell death, apoptosis. One is angiogenesis control. And I'll just look at that. These are untreated tumors. They're huge. They're full of blood vessels. And here are treated tumors. And those aren't full of tumor cells. Mostly that's just kind of what's left after the tumor cells are killed, the inflammatory cells, fibrosis, et cetera. It's just amazing. That's why we took pictures. This is the cytokine environment we're showing you that shows there's induction of a variety of inflammatory or immune pathways, including pro-immunogenic or, well, pathways that would cause the host immune system to attack the tumor. I'll just say again that we get this change in macrophages from tumor promoter to tumor killing, and an increase in T cells and macrophages into the tumor with the therapy that would be important for inducing that immune response against the tumor cells. And not to get into this too deeply, but amazingly, we saw an increase in killer T cells, the CD8 cells, and almost more amazingly, a suppression of what are called Tregs, or T cells that actually suppress the immune response and keep it cold. So like when you look at all these data together, it was like, I have to say as a scientist, a fantasy. And we couldn't have asked for more in terms of mechanism that support the therapeutic. I'm gonna walk you through these data very quickly and ignore some of it to keep it simple. Here's an experiment, and you can see kind of up here, we implant 2 tumors in one of these mice and only treat one with direct injection and look at the effect on both tumors. We also look at checkpoint inhibition, but I'm going to leave that out for now because of time constraints. The black line, again, is tumor just injected with the saline solution. And again, this green line, and these are the fast growing tumors, days, right, is the bacteria. So we showed exactly the same thing that I showed you previously, that there's a very substantial effect of bacteria. But now let's look at the tumor growing on the other side of the mouse that we never injected with bacteria. And I can tell you, we looked very critically. There's zero evidence that bacteria that we inject in the other side gets to this tumor. It just doesn't. So that's not what's going on. It's the immune system doing this. Black bar, untreated. I have a dotted line because this tumor wasn't injected with anything. And look at this, the dotted green line. Again, dotted because this tumor, we never touched it. And there's a substantial effect on its growth. Interestingly, probably even more than the tumor that was injected with bacteria. And that's a story for another day. That's very exciting that the untreated tumor, it's not untreated because the host was injected with, other tumor was injected with bacteria, but let's say the uninjected tumor, actually there's better tumor control than in the bacterial injected tumor. Very exciting response. This has to be a systemic immunologic response. It doesn't prove it's adaptive or specific immunity, though we have a lot of evidence that it is, but this will. In this case, this is called a rechallenge experiment. We start one tumor growing on one side of the animal, treat it with bacteria, right? We inject it on day 8, and then quite a bit later, day 20, we implant cells on the other side. In this case, we actually implant in the nerve, and they grow even faster there. And I did that because I wanted to, what I say, bias the experiment against seeing an effect. Black bar, saline injected tumors, controls. Again, green bar, the bacterial injected tumors, we see exactly the same effect I've already shown you. I'll just mention, when you combine with this checkpoint inhibitor, you get an additive effect. But again, that's a story for another day. But what about these tumors that are implanted well after this the bacterial therapy is done? Let's see what happens. Black bar, how they grow in the saline, previously saline treated animals. And look at this, the dotted green line. There's very, very little tumor growth. So the animal, and this almost certainly has to be adaptive immunity. There may be what we call innate immunity going on as well. The animal's able to control growth of a subsequently developing tumor. And again, the initial data would suggest even better than control of the primary tumor. Very, very exciting. I will just say we have some efficacy in other tumor types. including meningioma, both benign and malignant, as well as peripheral nerve sheath tumors. That's kind of our initial foray into NF1 type tumors. What's most interesting to me about these data is we've always seen the same thing. The slower the tumor is growing, the better the bacteria works. So here's the benign meningioma, slow growing, treatment with bacteria, black untreated. In this case, green again is their bacterial product you know about. You actually get regression of the tumor. When it's growing fast, this is malignant meningioma, you get control of tumor, but not regression. And we see the same thing with the NF1 type tumor, black bar PBS, slow growing or slower growing. And this is even faster growing than the human Schwannomas and tumors. We actually see regression. When they're growing a little faster, we see control. All very exciting data to us. And I will just mention all these data and other data that are associated with this have been published in PNAS, which is a very high impact journal. I will mention, as Neil did, that I'm working with John Mekalanos, who is the former chair of microbiology and immunology at Harvard Med School, and just a brilliant guy and wonderful part of the team to help us develop the next generation products. So we're very lucky to have him on board. And I am happy to talk to anyone offline about our data, to go do a deeper dive, talk about the things the data we haven't talked about today. Very briefly, I will just mention that, again, I don't know if I mentioned this, I continue to work with Neil and Mulberry. I'm on the advisory board. One of the questions was, if we make a bacteria in the manner that we would make it to inject in a human being, would it still work? And that's not a definite yes, because when we grow the bacteria for animal work, we add all sorts of growth factors to keep the bacteria healthy and strong that are derived from animal products, and you can't do that for injecting the bacteria in humans. So this is a critical experiment. We make a very similar product grown in animal-free media, and without belaboring this is the human Schwannoma model. That other product, which we're calling MUL-001, works absolutely as well as the animal product. And in the mouse Schwannoma model, same picture, works absolutely as well. That's the green line is the human product. The orange line is the older animal product. So this was very important enabling work. I will also tell you, we published this as well. We looked at the question, does the bacteria need to survive in order to have its effect? Because that's not a given. People have tried killing bacteria and then injecting them. But what we showed was if you inject the bacteria and then kill it a day later with antibiotics, trust me, that's the green line. There's no effect compared to saline injection. And this is our control, the regular, no antibiotics. But if you wait just 8 days and kill it, you do see an effect, albeit not quite as good as if you don't use antibiotics. I just mention this because I want you to know that we're doing the studies in animals in order to understand what will be necessary to treat these tumors in humans. So I don't know, let me see what time it is. I think I'm getting a little tight. Just to summarize, we showed that this bacterial approach with Salmonella works very well in both human Schwannoma tumors as well as mouse Schwannoma tumors. We've defined several mechanisms of action, all of which can be important for controlling and regressing these tumors. I'll mention that this isn't a kind of one-shot deal. We showed efficacy in 4 other tumor models, which I think is important for giving us confidence that this really could work in humans. And we've published in PNAS, which really in the scientific world is good confirmation that other people not involved in the project believe in our data. And I think that's all I have to say. Thank you.
Great, thanks, Gary. Really appreciate that. And as everybody could see, hopefully this was very, very exciting data. And you know, talking to Gary and John in the early days, that was really the draw for me into the company. It was the quality and extent of the data that was generated on the R side, on the research side. So what do we do with that now? And I don't want to go into too much detail on this, and I don't know how much everybody knows on the call about drug development, but I thought I'd just spend a few minutes explaining a little bit about, you know, how a drug is developed in the US. It's a highly regulated environment. You know, there's a lot of information that needs to be provided to lots of different people in order to try to extrapolate all the animal work that you saw into real patients. And so this chart here just gives you an idea about the phases. And we're right at the preclinical development phase right now, where we have data that shows safety and efficacy in animals, which of course doesn't guarantee that a drug is going to work in people. But what it does is that it allows us to have conversations with regulatory agencies, key opinion leaders, and others to make sure that we're doing the right thing to take the next steps into the clinic. You'll see in a few minutes that one of the most important parts of that development cycle is submission of an IND. And I have a slide a little later that's got all of these acronyms on. I know it can get a little overwhelming at times, but IND is an investigational new drug application. And this is a submission to the FDA in the US that allows you to proceed with clinical study. Typically, a typical IND is probably between 10 and 15 volumes of data, each volume being about 300 pages. So there's a lot of information to provide here. One of the most important parts of that is to go talk to the FDA before you submit the IND to make sure that they understand what you're doing, how you're doing it, to get their buy-in essentially before proceeding to the IND. Following the IND, you're allowed to initiate clinical studies. This is a chart that shows what is traditional drug development, where you progress from phase 1, which is typically healthy volunteers, to phase 2 to phase 3. It's a very long process. In rare diseases, because of the numbers of patients and so on, you know, it tends to be a somewhat truncated development plan. In fact, I've worked on phase 1/3 studies, so progressing all the way through to an approval study very, very quickly. So hopefully that gives you a little bit of a background. So when I talk about where we're at, you get an idea of where we are right now. Next slide. So this shows you some of the things that we've been doing since we raised the money and since the company was established. So we believe that as a result of a conversation with the FDA, the pre-IND meeting in September of 2021, we have a clear understanding about what we need to do to submit the IND and to proceed into patients. And that included the manufacturing piece. CMC is the chemistry manufacturing control section of an application. Essentially says how you're going to make the product. Non-clinical toxicology is a little bit different than the work that Gary described. It's traditional, how safe is this particular approach, this drug, when you inject it into animals. It's a pure safety concern. We also submitted to the FDA an overview. It was a fairly detailed overview, but an overview nonetheless about our clinical development plan. And you know, we got them to give us feedback and support for what we plan to do in NF2. We also applied for orphan designation, which is an important stepping stone in developing drugs for rare disorders in both the US and Europe, and they were approved very quickly. And we also are looking to see if we can perhaps get a pediatric designation as well. That's something that we're working on right now. So we progressed our manufacturing to the point where we'd be able to start GMP manufacturing, which means this is the material that's going to go into patients. We've defined our toxicology studies, and we've got the contract research organization available and ready to start those studies as soon as we have the drug available. I'll say a little bit more about the team in a few minutes, but we have a very, very high quality team. It's not just me, it's not just Gary. We have a team behind us that is developing MUL001. We've had great conversations under Gary's leadership with the Clinical Advisory Board to help us think about what are the right ways to study this approach in people. And so we've made a huge amount of progress towards filing the IND, hopefully by the middle of this year. And the idea would be that we would be able to start a phase 1, it would be a phase 1/2 study in patients by the end of the year. Next. So let me just say a little bit about the team. You know, Mulberry has one real full-time person working on it, and that's me. Everybody else who's engaged with this are consultants, contractors. It's virtual drug development. It's a process that I've been involved with many times over the years. It's a very cost-efficient and effective way to develop drugs because you get access to very, very experienced people with a tremendous set of knowledge to be able to help you develop the drug much more effectively and efficiently. And some of the names of the people you see there are people who have years and years of experience of working with regulatory agencies and manufacturing sites to make sure that we make this product as effective as we can make it. So we have a world-class team from that regard. I want to give a shout out, obviously, to the board. I think Susan might be on the call today. Susan and Phil, together with John and Gary, were the founders of Mulberry, and we were able, as Gilles said earlier, to bring in Tal Fura, who I'd worked with before very effectively on another rare disease for another company called Orphan Technologies, which we were able to take all the way through to the clinic. So we're also very fortunate to be working with scientific advisors and founders. Obviously, you've heard a lot about Mekalanos and Brenner today. Joe Kissel is a name that some of you may know we've been working with for some time. Caleb Yohe from the clinical side, as well as Brad Welling. Obviously, this is it's a relatively small group of experts who work on NF2, and we've been lucky enough to work with many of those people. I do want to say one more thing about Phil in particular. You know, Phil is a world-recognized orphan drug expert, and in fact, next slide. This is where I'm acting as Phil's literary agent. He wrote a very good book. He's written several books, but his latest is actually subtitled "A Quest to Save Children with Rare Genetic Disorders." It's a really good read. It explains all of the things that Phil's been involved with over many years, and I'd encourage you to take a look at it. So there's a link there if you're interested in buying that from Amazon, and Phil gives me a modest cut of his earnings there. Okay, next slide. This is another way to look at the timeline that we have in place. There's nothing different than I spoke to you earlier about. The goal is to submit the IND in the middle of this year, with starting the phase 1 study by the end of this year. So that's what we've got ahead of us. We're currently out raising money to make sure that this clinical study happens. And next slide. We'd be happy to answer any questions you may have. You know, with Gary and myself available, I think we should be able to cover most of the questions, but happy to answer any questions you have.
Thank you, Neil. Thank you, Gary. So yes, we have a few questions here.
Gilles, do you want me to stop sharing or keep this?
That's fine.
Okay.
Yes. So we have a few questions regarding vestibular schwannoma. So do you envision a treatment for vestibular schwannoma? And the follow-up question on that, saying that sometimes the size of the vestibular schwannoma is not always related to the loss of hearing. So it might reduce the size of the vestibular schwannoma, but what about the hearing? And also technically, will we be able to inject into a vestibular schwannoma? Or like you described earlier, saying that we don't have basically maybe to inject in every tumor, it will have an impact on the other tumor. So maybe you could first answer the questions specifically about vestibular schwannoma, and then there is other questions.
Neil, do you want to take it or do you want me to start?
Yeah, I'll hand it over to you. As I
Okay.
mentioned in my presentation, Gary has been leading the effort on the clinical design. And maybe you can say a few words, Gary, about where we're starting with
Sure.
regards to that clinical study in the peripheral tumors.
So I would start by saying, I don't think that any therapeutic that couldn't target vestibular schwannomas would be an important therapeutic for NF2 patients. So of course, our lab always keeps that in mind, that our goal is to target not only peripheral tumors, but intracranial tumors. And don't forget the perispinal tumors, which, I'll just mention, I got interested in working in this area because I see patients with pain from NF tumors. So I have many, decade plus, 20 years, whatever, experience with patients with NF disease. So the fast answer to the question is yes, we envision a pathway to treating intracranial and perispinal tumors. And we've talked to multiple neurosurgeons and ENT surgeons early on, like, would you be comfortable doing this, both in terms of getting to the tumors as well as injecting an attenuated bacterium? And actually, I was shocked because everyone we spoke to said, no problem, that we'd much rather be able to access a tumor and inject it than access a tumor and resect it. We feel that that would be much safer. As Neil kind of alluded to, we wouldn't start with injecting intracranial tumors. When our clinical design, for concern about patients' well-being, because we never want to make someone's life worse with therapeutic testing, is our initial trials would all be in adults and would target peripheral tumors. So distal arm, distal leg, not involving major neurologic studies like the brachial plexus under the arm, where you can find tumors. To get the first question we want to answer is it safe in humans? And get some information about the interaction between the bacteria and the tumors in a human rather than a mouse. Now, the second question about hearing loss, that's actually a very interesting question. I'm a pain guy, and much of my research was previously in the pain space. And we actually published a study on the mechanisms of pain with schwannoma. And just like hearing, which is what we call a gain of function, actually hearing is loss of function, sorry, tinnitus, which is gain of function. Pain we think of as gain of sensory function. These pain fibers are being activated by these tumors. And if you look at the clinical data, while this can happen by just growth of the tumor and compression, there's plenty of reason to think that's not the only way that happens. And I'll mention my PhD. I was actually an immunologist and a neuroscientist. So I had a kind of inkling of how important the interaction between the nervous system and other cell types, including Schwann cells, is. And there's every reason to believe, just like pain, which you can go online and see our study on pain mechanisms of schwannomas. And we looked at human tumors, by the way. We showed that it's not just a size phenomenon, that we actually identified a substance these tumors release and cause pain. And the reason I'm bringing this up is it's very interesting. Some of the therapeutic strategies can actually help with pain, even in the absence of decreasing size, we believe. So we do think that it's possible that a therapeutic that debulks tumors by kind of like killing tumor cells and making other tumor cells sick in a way could actually help with some of these other sensory problems. And it's not just hearing loss and pain. There's also balance problems, as you're aware of. Tinnitus can be a big problem for people. So we're hopeful that the treatment can help with these sensory deficits, whether it's a gain of function, like pain, or a loss of function, like hearing loss. And while naturally, my hope would be you could treat a peripheral tumor and it would lead to regression of other tumors throughout the body, I think we have to be, in a sense, I'll call it practical, and at least assume that we have to treat all tumors to get a direct effect, but still hope that that could have effect on other tumors or development of subsequent tumors. So our developmental pathway always assumes that we should be able to inject any of these tumors. Sorry if that's a long-winded response.
Thank you. No, but that's good. You answered also to a few other questions at the same time that were posted. A few questions about the immune response. So, question from Nicole, that she's next to you, I guess. How do you expect the immune system of humans to respond compared to the immune system of the mice?
Yeah, so I will say that mouse and human immune systems are different. Having said that, the data we have related to human tumors does suggest that they very likely represent an immunologically cold environment. The product that's used to treat bladder cancer, and again, the bladder cancer that's being treated is a slow growing cancer that hasn't, it is a cancer, but it's slow growing and it's only used when it's non-invasive. So I would call it a cancer that's still in a benign-like phase. So I find it very interesting that the one bacterial therapy that works for a cancer is a cancer that's close to a benign-like tumor. And the data we have suggests that there's a significant inflammatory component to how that therapy works. So those are reasons I think that we may see similar immunological responses to bacterial treatment in humans as we see in mice. Again, to be kind of practical, we are thinking ahead about whether or not some sort of multimodal approach like checkpoint inhibition, and we are thinking of others, might be helpful to enhance the pro-inflammatory effect of the bacteria. But remember, they're not only killing by inducing anti-tumor immunity, there's also direct killing, anti-immunogenic effects, vascular effects, so angiogenesis effects, as well as induction of innate, so not the specific immune responses, but innate immune responses that could also be important for tumor control. So that's one of the reasons we want to get to human trials so quickly, is to get more information on what will occur in a human being.
Thank you. We saw on your slide that you also injected checkpoint inhibitor PD-1. So we have a question from Eric about it. If you are planning to use PD-1 inhibitor in one of the phase study, or will you do it later and restart the whole process of clinical trial?
So I'll just speak for myself. My hope is the bacteria alone, like the BCG product, which has a cure rate, a stated cure rate of 60%. And remember, a lot of the folks who aren't cured have tumor control, and they just have the BCG therapy repeated every several years. That's not so terrible either if it keeps a person healthy and alive. So my hope is bacteria alone are adequate as a treatment, but we also, I don't like the term hedge our bets, but we want to think ahead to think about how we might even make the treatment better. And I think immune modulators are an obvious approach, given that we think a major way this bacterial therapy is going to work is through activation of immune responses. So what can we give with the bacteria or following the bacteria to maintain the changes that the bacteria induce?
Yeah, I think just to reiterate that, I think the important thing is that we need to establish that bacterial therapy as a standalone therapeutic modality has some benefits and reflects the mouse data that's been studied so far. So, we may move on to other things later, but we've got to establish that bacterial therapy works in benign tumors.
And again, I just say that the bacterial therapy used to treat bladder cancer, it's not a combination therapy. It's bacteria alone.
Yeah.
Okay, thank you. Question about the trial. How many people in each of these trial phase are you planning or expecting?
So let me take that one, Gary, if I can. I showed that chart that showed phase 1, phase 2, phase 3. That isn't necessarily how this approach is going to be developed. As I said, in rare disorders, rare diseases, it's usual to take somewhat of a different approach to the phases of clinical study. As I said, phase 1 tends to be healthy volunteers. If I was developing another non-steroidal anti-inflammatory drug, first study would be in healthy volunteers. We're not going to do that here. We will go straight into patients with NF2. So there's already some differences in those phases. So just keep that in mind. Same thing for the numbers of patients. If you were asking me how many patients do I need in a pivotal phase 3 trial for a non-steroidal anti-inflammatory, I could probably give you a number immediately in the thousands, right? However, with rare diseases, and having been developing these drugs for 30 odd years now, it depends. It's a terrible answer, I understand, but you've got to be driven by the data. And there are things we will learn in this phase 1/2 trial that will inform exactly that. What are the right endpoints? How long will we study patients for? How many patients do we need to show an effect? Of course, the bigger effect that we see, the smaller the number of patients that we'll need to study. So it's not a great answer because I can't give you an exact number. The first study is going to be somewhere between 20 and 30 patients. And that also depends upon the data generated during that study, because it's a traditional design where we will increase the dose gradually. And we may decide at some point to stop that. Or if we see some other effects, we may decide to stop the study at a lower dose. It all depends upon the data. There's just jumping through the questions, Gilles, because I know that we're running a bit short on time. There was a question about what have been the challenges with bacteria therapy to date. And I'll speak from my perspective as a drug developer. The big problem with bacteria therapy, other than BCG, is they haven't shown any efficacy data. Or they haven't shown much efficacy data in people. Remembering that bacteria therapy has never been tried in benign tumors. All of the experience to date has been in oncology setting, which, as Gary mentioned, is a very different environment, very different tumor environment. And we believe that we understand why our approach in benign tumors is potentially going to work, whereas other bacteria therapy approaches didn't. I want to remind people also that we have a very simple off switch, a kill switch, if you like, which is if we see any problems during the clinical study, and in fact, it's built into our study design, you can treat these patients with an antibiotic, which immediately will kill the bugs. wherever they may be. So it's not like gene therapy where you integrate into the DNA. This is not that. This is a therapeutic immunotherapy, and you can switch it off.
Thank you. We have many more questions that we have 3 more minutes, so we can maybe take
Sure.
1 or 2. And then the rest of the questions, I will collect them and I will share them with you, Gary and Neil, and I will make
Sorry to interrupt. I just wanted to interject one more point, that this product has been in multiple trials, and I alluded to this, with intravenous delivery, and it was well tolerated. So we have every reason to think intratumoral delivery will be very well tolerated. And there was one derivative of this bacterial product where they actually added something to the bacteria, something called cytosine deaminase. But in that trial, the bacteria were injected directly into tumors. And again, it was very well tolerated. So we don't anticipate that the treatment would be limited by toxicity.
Questions from Mary. As you know, many NF2 patients are trying drugs, taking Avastin or are currently on other trials of other drugs. And so the question is, will trial participants, from your experience, Neil, from previous rare disease that you worked on, what about the people that are on drugs, on other drugs, or did other trial drugs? Do they need to stop before starting this drug trial, or because they tried something else before, they cannot be eligible to this new trial that you are planning? If you could clarify that for us. Thank you.
Yeah, again, just to be really specific here, we haven't finalized this protocol. We've spoken to the FDA about the overall design, the kinds of endpoints, the kinds of patients. We got some great feedback from them. We're integrating that into the next version of the protocol. There are still some specifics that we're still working on. So that's the first point. There is no final protocol. We don't know exactly, where the trial sites are yet. We've spoken to several places where we could do the study, all in the US. That was another question that I think we saw. Is this US only? At this time, yes, it is. It's US only. And that's from a regulatory perspective. So, we're still working on some of those aspects. I'm sure Gary has some much better answers to this, but, we'll know more once we filed the protocol, the full protocol to the FDA, and had formal feedback in the IND.
Yeah, I would just say that every trial has inclusion and exclusion criteria, and you really have to be very thoughtful, because you don't want to develop a product that works for a very small subset of patients because of the people you let in was so limited. But you also don't want to erroneously kill a product because you're letting the wrong people in and you erroneously conclude it's not efficacious or it's unsafe. So as Neil said, we've been working with really smart people from around the country to think of how to do this in the optimal manner. To give you one example, we probably wouldn't enroll patients who are currently on any sort of immunotherapeutic agent for obvious reasons, because we'd be concerned that that might interact with what the bacteria need to do. And that's all I'll say. I just want to give you one example of what might exclude someone from the trial. Immunosuppressed individuals would probably not be in a first phase trial, early phase trial either.
Good. Thank you so much. We are on time, and this webinar will be recorded, and we will advertise the recording on Facebook and on in our newsletter, and it's accessible on the YouTube channel. And we'll collect the remaining questions and share them with Gary and Neil, and make sure they are answered as well.
And Gilles, thanks. I just want to thank you again for the opportunity to present, and also for the critical support that NF2 BioSolutions provided to our lab to really keep the project moving forward after we made the initial observations.
Yes, indeed, it's a real challenge. As a nonprofit, we are capable of jumpstart of supporting preclinical research, but then to move through that phase of preparing a trial is a totally different ballgame. And we are trying to do our best to help you, Mulberry, you, Neil, and Gary to move forward with it. Like you are one of our closest chance to have a long-term therapy for our kids, our patients. Thank you so much again, and hope to see you again for great new updates.
Thank you. Appreciate it.
Thank you.